Okay, so I would like to talk to you a little bit about chromosome mapping. So we're going to talk about both chromosome mapping, the process, and taking advantage of the chromosomes that you've mapped. Creating this map and then ultimately using it to learn new things about your matches. So kind of our roadmap here is first we're going to talk about the concept of chromosome mapping. Then we will talk about finding DNA to add to your map. And then we'll talk about utilizing that map once you've gone about the process of creating it. And so when we talk about chromosome mapping, those of you that have done this are familiar with it. But if you're new to chromosome mapping, it's essentially a technique we use to assign shared DNA to a particular ancestral couple. Now, so what we're doing is we're taking existing DNA matches and we are discovering what DNA we share with them and assigning it back to the ancestral couple we share with that particular cousin. So to do chromosome mapping, it's essentially a three-step process. At the first step, what we have to do is we have to find a known cousin. So we're not going to map unknowns because we don't know how we're related to them. We don't know what ancestral couple we share with this unknown. So we have to have some known cousin. We have to find the DNA that we share with that known cousin. So in some cases, it'll be one single piece of DNA. In other cases, it will be a lot of DNA. But we have to identify what those pieces are. Once we have that information, we can then proceed to adding those pieces or piece of DNA to our existing map. Now, when I talk about a known cousin, it has a very specific meaning. It means that we know who they are, so we know who the person is, and even more importantly, is we know how that person is related to us. So when we talk about how they're related to us, what that means is we've identified the genealogical pathway between that person and ourselves. Now, most of the time, hopefully, that's a simple single pathway, right? From one ancestral couple down to us. Unfortunately, as we know, we have cousins where we're related to them several times, which makes chromosome mapping a little bit more difficult. Not impossible, but more difficult. So if we can find a known cousin and we have only a single genealogical pathway, that's going to be where we want to start with our chromosome mapping. So for step one, for example, if we're finding a known cousin, this is Ellen Jones. This is one of my MyHeritage matches. Now, there's not a real Ellen Jones. All the names throughout my presentation will be obscured for privacy or changed. But let's say this is my cousin Ellen Jones. You can see she shares about 66 centimorgans with me there on the graph, on the chart. Now, I know through traditional genealogical research that Ellen is related to me through our shared great-great-great-grandparents. So that means that Ellen is my fourth cousin. Okay, so I now have a hypothesis about our shared DNA. So because we have this known genealogical relationship, my hypothesis is that the DNA we share in common is from our shared ancestry. And so it comes from our great-great-great-grandparents. So whatever pieces or piece of DNA that we share came from that ancestral couple, one of them, we don't know which it came from at this point, down my line to me, and from that ancestral couple down her line to Ellen. Okay? Now, that's step one, right? Step one is find a known cousin with a known genealogical relationship. Step two, then, is finding what piece or pieces of DNA that we share. Thankfully, MyHeritage has a chromosome browser that gives us what's called segment data. And segment data is the DNA that Ellen and I share in common. I often think of it as being GPS coordinates. So it's GPS coordinates, locations on our chromosomes of where we share DNA with Ellen. And so the coordinates requires a couple pieces of information. First of all, I have to know what chromosome it is on. Is it 1, 2, all the way up to 22? It has to be on at least one of those chromosomes. And then I have to know where does it start on that chromosome and where does it end. Now, in addition to that, some of the optional information that's usually included in there are things like how big is that piece of DNA? If you were here for the previous talk, you heard talk of centimorgans, and that's how the distance is measured in centimorgans, kind of a length of DNA. And then how many positions were tested inside that segment? How many SNPs were analyzed? And this is essentially the segment data. So if we look at a chromosome, for example, like this one, The piece of DNA in orange there is the shared segment, say, between me and Ellen, and the pieces of information I need to be able to put that into a chromosome mapping software is, what chromosome number is this? And you can see this is chromosome number 2. And it has some starting location and some stopping location. And that's what we can get from the chromosome browser at MyHeritage. Now, we can get that information in 2 different ways. So there are actually 2 different approaches to get segment data, the GPS coordinates, from the chromosome browser at MyHeritage. And I'll show you both ways to do that. The first way to look at the chromosome browser at MyHeritage is through the DNA Tools portal. So if we go to the main page at MyHeritage here, up at the top there is the DNA button. So you can press that and you'll get the drop down. And then under that we have the DNA Tools button. Now, there are a number of different tools under here. You can see there is a chromosome browser. There is the auto cluster tool, which we'll talk about in a few minutes. And there also is the ethnicities map. For this one, we're going to go to the chromosome browser. And when we look at this, we have the option now of selecting different people from our matches to compare in the chromosome browser. So let's say you have a couple of known cousins at MyHeritage. You can select them with this tool. You'll see you can do a search. You can find those cousins. And then when you select them and you hit compare, you'll get something that looks like this. So here what I'm doing is I'm comparing 5 people against my chromosomes, and the colors correspond to each of the individuals. So we have yellow, red, tan, green, and so on. And you can see on the actual chromosomes there, 4, 5, and 6, those are the colors that represent the pieces of DNA that we share. So now if I want to get the actual GPS coordinates, you can see I don't actually have those there, right? I have the chromosome number. So if we look at the red individual, you can see we have a couple of large pieces, a large piece on 4, a really, really large piece on 6. So that's the initial number. But I have to find out, where does that piece start? Where does that piece stop? So if I scroll down to the bottom of this page, I'll have this additional interface underneath the chromosome browser. And here I can click on any one of these individuals, and then that will give me the GPS coordinates for that particular individual. So here, I'm going to do that for person number 1. And when I click on the little drop-down icon, I get a full list of all of the pieces of DNA that we share. And this is the GPS coordinates. So we have a bunch of different pieces, 1, 2, 4, 4, 4. Those are all the chromosome number. Then you can see we have a start location, a stop location. Towards the end, we have the number of centimorgans, or the size of that piece, and then the number of SNPs that were tested. So that's one approach. You can go right to the DNA tools, down to the chromosome browser, and compare multiple people at once. I think probably the more traditional approach to get that segment data is by getting it from each of the individual match pages. So in the previous slide, when I was looking at those 5 individuals, I was looking at this view all at once. But I could go to each one of those 5 individually and get the segment information from that individual page. So here I would go to, say, Ellen's page. Right? Ellen, remember, she is my 4th cousin. So when I open Ellen's page, here I have the option now, I will scroll down, and on the bottom of every match page, of course, is the chromosome browser that shows the pieces of DNA. So when I scroll down on Ellen's page here, I now have the piece of DNA that I share with Ellen here on chromosome 5. You can see that purple piece, right? The gray is the empty chromosomes. We don't share any DNA there, but we have the purple piece where we do share that particular chromosome. So when I click on that particular piece, up comes a pop-up with that segment information, with the GPS coordinates that I need to be able to take this from here and put it into software where I can keep track of all of these pieces of DNA. All right, so here, for example, it's on chromosome 5, right? We have that same piece of information, the chromosome number, where it starts, where it stops, the size of the piece, and the number of SNPs that were tested. So remember what our hypothesis is for this particular piece on chromosome 5. Our hypothesis is that since we both share this piece of DNA, that piece of DNA came to both of us through each of our individual lines and is a piece of DNA that our shared ancestors shared in common. right? Or our shared ancestors possessed this piece of DNA. The problem is that we don't know which ancestor this piece could have come from. It could have come from either of them. So when we get to this point, for example, if I'm going to record this particular piece of DNA in any sort of mapping software, I would give that piece of DNA the name of both of those ancestors, right? So if we say the people in the red box there are Jane Smith and John Doe, for example, this ancestral great-great-great-grandparents, I'm going to assign that piece of DNA to give it that name, right? Now, down the road, what I hope is I'll get a match that's even further back than one of these two that share that piece of DNA, and then I can assign that piece of DNA back to that ancestral couple. And that's one of the goals of chromosome mapping. You'd like to, for example, have all of your pieces of DNA mapped to your grandparents, and then go back another generation and figure out where they came from the great-grandparents and so on. So we want to do that and work our way backwards, filling in more and more segments as we go. So now that I have this segment, now that I have the GPS coordinates, at this point, now I can go and I can add this to any sort of mapping software, keeping track of these pieces of DNA. One of the advantages of chromosome mapping, in addition to the ones we'll talk about, is it's a great organizational tool. It's a way for you to get this segment data and keep track of it in one place. Now, you could always, at MyHeritage, one of the options is you can actually download a spreadsheet of every single piece of DNA that you share with every single one of your matches. Personally, I'm not a big fan of spreadsheets. I try to avoid them whenever I can. It's not always possible in the world of DNA. Often you're just going to be stuck with spreadsheets. But for me, keeping track of this information in software where you're actually mapping these pieces and where it's visual and retrievable is much better than a spreadsheet where you're always, it's just too abstract. I think visualization of these pieces of DNA is really key to using them. One of the most common questions I get when it comes to chromosome mapping is who we should use. And the answer to that really is almost anyone. Of course, remember, we have to satisfy that first step. It has to be a known cousin, which means it has to be a known individual with a known ancestral connection. That genealogical path between them and us has to be known. So if you satisfy that connection, then you can use that person for mapping. With some exceptions, I wouldn't map a parent because you share full chromosomes with a parent, and so that's not going to help you out with your map. Same with siblings, because what lines do you share with your siblings? You share every single one of your lines, for example, with a full sibling. So adding a sibling to your map isn't going to be helpful. Some of my favorite relationships to add to a map are these, although there are several in here I can't do. I've no tested grandparents, for example. I can't test great aunts and uncles. I don't have any half aunts and uncles. Now, I have lots of second cousins. And second cousins are probably my favorite cousins to map for two reasons. The DNA you share with a second cousin goes all the way back to your great-grandparents. So what that means is every time you find a piece of DNA you share with a second cousin, you can assign it to those those great-grandparents that you share with that second cousin. The other reason I like second cousins so much is because they share, on average, more than 200 centimorgans with you. It varies. Maybe 175 to 250 is the biggest range usually. But that's a lot of DNA to share with someone and to assign back to a great-grandparent level. So they're a really good connection. Now, even better than that is a half-first cousin, because they also go back to shared great-grandparents, and they actually share twice the amount of a second cousin. But since I don't have any half-first cousins, they're not my favorite. But if you have half-first cousins, they should be your favorite. Now, second cousins, half-first cousins, third cousins, these are all perfectly fine relationships. Also the same when we get to removed, right? So the child of a third cousin would be your third cousin once removed. That's perfectly fine to map as well. When we talk about how much DNA we're getting from a particular relative, this is a comparison of 3 different relationships to give you an idea of how much DNA you're able to map when you can see relationships with one of these particular cousins. So here we're looking at a great uncle, a second cousin, and a fourth cousin. So with a great uncle, there's a total of 33 segments of DNA that you actually get to assign back to that ancestry you share with a great uncle. So something like that would be a fantastic relationship if that's a possibility. It's not for most people, but for people who it is, it's a great relationship. You can see all the red segments on these chromosomes. Those are all segments you would be able to assign back to the great-grandparents that you share in common with that great-uncle. Now, the more distant the relationship, the fewer the pieces of DNA we're able to assign. However, in the orange there is a second cousin, and you can see that is 16 segments. So that's still a lot of DNA that you're able to assign back to great-grandparents. And then we get to a fourth cousin, which is in yellow, and it happens to be just one piece of DNA on one chromosome. So you can see it's a nice piece of DNA on chromosome 5, but that's all the DNA you're going to be able to assign for that particular relationship. On the other hand, think about how far back in time you're actually assigning this piece of DNA, right? This piece of DNA is going to go back to great-great-great-grandparents. And so that's a really far back in your tree to be assigning DNA, to know that you actually received this piece of DNA from that particular relationship. Now, that being said, I want to caution you about these distant matches. So fourth cousins, fifth, sixth, and so on. You want to be careful about mapping those relationships because there's the likelihood that you could be related on a line other than the one you're aware of, right? You may think, well, I know I'm related to this person, for example, when it comes to a fifth cousin. I know we share great-great-great-grandparents, and so I feel comfortable assigning to that relationship. On the other hand, if, say, the other person's tree or your tree isn't very complete, there's always the possibility that pieces of DNA could have come from some other relationship that you don't know. And this is more important in areas that don't have trees or records as complete as in other areas. So, for example, in the Netherlands, I know you have great records, so maybe this isn't as much of a problem. Whereas the other issue may come in, such as having relationships on more than one line with a particular cousin. But those are both situations where you have to be careful. Now, on the other hand, if I were to say find a piece of DNA that I share with a fourth cousin once removed, or twice removed here, it's a small piece, 12 centimorgans. It's the only piece of DNA that we share. I wouldn't hesitate to add this to my map. In fact, I consider all of the pieces of DNA that I add to my map to be tentative. Because you never know if some new piece of information is going to show you that, sure, you map these pieces of DNA to this relative, but it turns out they're not just your third cousin, they're also your fifth cousin or some other relationship. Now, the nice thing about chromosome mapping is the more and more pieces of DNA that you add to your map, the clearer it's going to become when you make an error in an assignment. So, for example, I mapped someone who is my fourth cousin, and there were 3 pieces of DNA, relatively small pieces, and I all assigned those back to our shared ancestry on my father's side of the family. Well, I continued to add more and more pieces to my map, and it became very clear that one of those pieces didn't behave the same as the others. It didn't overlap properly with that paternal side that I assigned it to. And it turns out that 2 of those pieces came from my father's side, and 1 piece comes from an unknown maternal relationship that I actually share with this match. So every piece of DNA you assign should be tentative, and realizing that down the road you may have to change it from one line to another. Right? So getting these pieces of DNA, the more people you're able to identify among your matches, or the more relatives you ask to test, the more pieces of DNA you're going to be adding to your map and utilizing for mapping. Right? So one of the questions is, why do we go to all this trouble? On the one hand, I think it's fun. It's really an enjoyable, I find it almost relaxing to go in and start adding these maps, these pieces of DNA, and creating this beautiful, colorful visualization of your DNA. But on the other hand, we're all busy people, so there should be some sort of direct benefit from this type of mapping. So there are a couple of things that once you start to build a map, you can then use it to learn things about both new and existing matches that you have. So first, I want to talk about characterizing new MyHeritage matches using your existing chromosome map. Now, the more complete your map, obviously, the better this particular process is going to work. So if you're just starting off with your map, this may not be something that's too successful. But once you start adding more and more and more pieces, hopefully you're going to be able to use it for more and more applications. So here's an example. I was looking among my matches, and I found this particular individual, and you can see they're from the Netherlands. So one of my goals here is to potentially use my map to figure out what line I share with this particular individual. You can see there's a tree with 33 people in it. So the first thing I always do with a match is, of course, review their family tree, because hopefully I find a family or a location or some line that jumps out at me and presents a hypothesis about how we're related. So, for example, if they had Bettinger in their family, that would be an easy one for me to focus on. Now, I opened up this tree and didn't recognize a single person in that tree. No surnames, none of the locations, and so that wasn't too fruitful. On the other hand, often what happens is you'll open up the match and they'll have no tree whatsoever, right? And then that option is completely not an option. So what I'm going to do here is I'm going to use my chromosome map to potentially see how I'm related to this individual. To do this, it's sort of a two-step process. So I'm going to open up this page, right? I'll click on New Match. I will open up this page, and the first thing I'm going to do is scroll down to see the shared matches that we share in common. All right, so when I look at the Review the DNA match and I scroll down to see the shared matches, I can see right away that this is, I believe, a maternal match. Now, how do I know that? Well, when you look at a shared match group, and shared matching is probably one of the most powerful tools we have as genealogists today. When I look at a shared match group, what we're trying to find is someone in that group we recognize. Because the hypothesis behind a shared match group is that we share, all of us, share some common ancestry. And that's why we are a shared match group. And so if you see someone in a shared match group that you recognize, what that says to you is this is probably the reason you have shared ancestry. Right? And so in this one, I recognize Belinda Walters, meaning she's a known match. So when Belinda Walters shows up in a shared match group, I already have a hypothesis about how that shared match group is related, because I know what my relationship to Belinda Walters is. For example, she's a maternal first cousin. Now, a maternal first cousin is great, but they don't push us back too far. Because what do I share with a maternal first cousin? I share my maternal lines, right? Essentially all of my maternal lines. So when you see a maternal cousin like this, a first cousin, in your shared match group, the best you can really hypothesize from it is that this is a maternal match. So that's step one, right? Step one is see if you can determine anything from the shared matches. And here, I don't learn much other than I know what side of the family she's probably related on this new, or he is, this new match from the Netherlands. I hypothesize now he's a maternal match because of the shared match group. So now that I have that information, now I go to step two, which is I scroll down on the page further to see the chromosome browser, because I want to find the piece or pieces of DNA that I share in common with this new match. And when I do that, I find that we share a almost 20 centimorgan piece of DNA on chromosome 22. That alone, of course, doesn't give me any useful information. It's only when I take that piece of DNA and align it with my existing chromosome map. Because what I'm looking for is I'm looking for overlap, right? So I'm going to take my chromosome 22. So this is what my map of chromosome 22 might look like. This is what we did, I was just talking about, right? I found a known match, I found the piece of DNA, and I added it to my chromosome map using any type of chromosome mapping software. So here, on my paternal chromosome, I've assigned a huge piece of DNA to my ancestors, John and Jane Smith Doe. That was on the basis of finding another descendant of John and Jane Smith Doe and figuring out we share this large piece of DNA on my paternal chromosome at this location. I also have a smaller piece of DNA that's mapped on my maternal chromosome. I found a maternal match related to me through Ed and Susan and assigned this piece of DNA to chromosome 22. All right, so what am I going to do? I'm going to take that piece of DNA that I have, that I share in common with that new Dutch match, and I'm going to overlay it onto this map. So when I do that, it shows up right at this location of my map. Now, simply on this basis, I can make an instant hypothesis, right? I have to be related to this Dutch match through either John and Jane Smith Doe or Ed and Susan Walters. Those are my only two options, right? Because I only have two chromosomes, a dad chromosome and a mom chromosome at this location. And if I have both of those mapped, meaning I know what ancestors I got each of those from, then I can narrow it down. I have to be related to this Dutch match through either Jane and John Smith Doe or Ed and Susan Walters. But because of shared matching, I can actually do better than that. Because I know from shared matching, this is a maternal match, right? My maternal first cousin showed up there, so I'm hypothesizing this is a maternal match. So what does that tell me? That tells me that, so remember we had Belinda in there. That tells me then that this has to be a maternal match. That also means I have to be related to this individual through Ed and Susan Walters. Now, so based simply on shared matching and on the location of this DNA and my existing chromosome map, I now have a hypothesis about how I'm related to this particular match. And so if I have any knowledge of the tree of Ed and Susan Walters, for example, now I can contact this match and hopefully make some sort of connection. Because in my experience, when we contact a match and we have meaningful information in there, including simply maybe just as simple as we have shared ancestry on this line, or it looks like you're related to me through this family based on shared matching. Here I can actually say, I'm related to you through the ancestry of Ed and Susan Walters, and I can share my tree and point that out. When I do something like that, that tends to significantly increase the response rate from that cousin. Years ago, when I first started in DNA, most of the communications I'd receive would be along the lines of, we show up as a match, would you like to share information? And I mean, yes, I would, but that's a really tough thing to respond to, because if you get so many of those, you don't know where to start, right? A much better communication, and those that I tend to receive now, are things like, it looks like we're related through X and Y, here's a link to my tree. What I'm finding is that today, people are so much more experienced with DNA, they tend to write more experienced communications. And when someone says to me, it looks like we're related through this line, here's my tree, it is an order of magnitude easier for me to go and look at that tree and examine that, versus where I have to initiate a relationship and say, yes, I would like to share information, here's my tree. So when you can do something like this, shared matching, your chromosome map, or whatever, and you can write better communications, you're going to tend to get better communications back. Now, I will not promise you that you're going to have everyone will respond and you'll be able to find the breakthrough, because we know better than that. But it increases the likelihood we're going to get a good response from that particular match. So, using your existing map is a good way to characterize new matches as they come in. Would I add this new Dutch match to my map? I wouldn't add them, because I don't yet know what the genealogical connection is. I might know who the person is, and now I at least know what line it's on, but it's not until I can actually assign this piece of DNA to the very ancestors that we share in common that I would actually add this to my map. That being said, people have different maps. I have, for example, what I call my gold standard map. That's the one where I only put in known relatives with a known genealogical connection. On the other hand, people also have tentative maps where they will be a little bit more flexible. They might add something like this, saying we share through this line, and they may not know the exact ancestor, but it's all dependent upon what kind of map you might want to have created. One of the other things that I find these chromosome maps very useful for is actually characterizing shared match clusters. So these shared match clusters at MyHeritage are called autoclusters. And so when you combine an autocluster and a chromosome map, you can actually gain some extremely valuable information about that particular shared match group. So I'm going to go really quickly through the process of creating these autoclusters, because they're a tool that's available at MyHeritage, and there's some instructions there. But to get to this tool, we're going to do the same thing we talked about before, going through the tools at the drop down here. So we'll go back to the main page, the main DNA page. We'll click on DNA, and then we click on DNA tools. And what we saw earlier is this is one of those tools that's there. We looked at the chromosome browser. This is now the auto clusters tool, and we also have the ethnicities map there as well. When you click on the auto clusters tool, you're going to see an option to generate auto clusters within MyHeritage, right? This is all looking at tools within MyHeritage. You'll have the option of creating an auto cluster for any of the DNA tests you've done through MyHeritage. So, for example, I've tested myself at MyHeritage and several different family members, so I can run an auto cluster for any one of those individuals. I'm going to run an auto cluster for myself, and then I can use that result. So when you click this auto cluster, what's going to happen is it is creating a chart, a diagram of your shared match groups. So what you can do is, as you probably know, shared matching, again, most powerful tool we have, I think, is we will click on a match and we get a list of the matches we share in common. So if I test myself and I find Cousin Jill, I'm going to get a list of all of the people that we share in common with Cousin Jill. And that's going to be a shared match group. So the purpose of auto clusters is to go in and grab a bunch of these shared match groups and organize it into a map. And what it looks like is something like this. Now, I'll receive it via email. So when I click that generate under the auto clusters option, I'm going to receive this email, and then I'll open it up. And so each one of these colored box groups is a different shared match group. So here in this particular analysis, I have 16 different shared match groups. And now the hypothesis between these shared match groups is that each group has common ancestry. So for example, everyone in the red group has common ancestry, in the orange group, and so on. So unfortunately, or fortunately, depending on your view, we're not at the point where the tool says this is your shared ancestry. Okay? So that's our goal, right? We want to figure out what is the shared ancestry, who is the common ancestor that's responsible for the formation of the red group, for the formation of the orange group, and so on. And then that will give us all sorts of information about the people within that group. So let's say, for example, I recognize a bunch of people in the red group, and I know exactly how we're related. That is essentially what we talked about with Belinda, right? I know, for example, I'm related to the individuals I identify in the red group through my great-grandparents. So what does that tell me about everyone else in the red group? That then says, I hypothesize we're all related through some line that's above my great-grandparents. That explains what our relationship is. So I want to do that for every single one of these because it's going to help me characterize a whole bunch of my matches at any one time. So what I'm going to do here is we're going to look at cluster 5. That's that blue cluster there. For me, this is a nice visualization, but that actually isn't where the information is. The information I want is actually down below this report. When you go to the table down below, this is where the real information of your auto cluster is found. These are the 5 people, the 5 matches I have in cluster 5. Again, what's the hypothesis? We all share common ancestry. If I recognize any one of these 5 individuals and I know what our genealogical relationship is, then I have an instant hypothesis about how we're all related. For these particular 5, I don't have any sort of hypothesis. I have no idea how I'm related to any one of those 5 individuals. So what I'm going to do is I'm going to open each of those matches. The nice thing about the auto cluster tool, because it's been integrated into MyHeritage, I can just click on any one of these names and it will open up in a new tab. So just go through and open up those 5 individuals. And for each one of the matches, what I'm going to do is I'm going to scroll down the page and go to the chromosome browser for those matches. And often, not always, but often what you'll find is there's a single piece of DNA or maybe several pieces of DNA that's shared by the individuals within that group. And in fact, when I looked at this particular group and I scrolled down the page, I discovered that every single one of them has an overlapping piece of DNA here on chromosome 20. So I'm obviously more closely related to some than others potentially because it's a bigger piece or a smaller piece. And so what I can do now is I can use this and my chromosome map to actually characterize the entire cluster. Because here, what do I have? I have a piece of DNA on chromosome 20. If I can take that and I can overlay it with my existing map, I might potentially learn information about this cluster. So that gives me kind of the rough location. I took the 2 farthest edges of these overlapping pieces. I'm simply going to use that as new GPS data, new segment data, and I'll go into the mapping software, and I'm simply asking the question, does this piece of DNA on chromosome 20 overlap any of the assigned segments that I already have on my map? And so I'm going to do that for chromosome 20. Here are the pieces again that I've assigned on this particular chromosome. Another piece from Jane and John and Jane Doe, for example. And on the maternal chromosome is JJ and Susan U. James. Right? So now what do? I take that piece of information, I overlay it here, and I have an instant hypothesis. This person has to be related to, or this cluster has to be related to me through one of these two lines, right? That's the only option. That's the only way for someone to be related to me in this location of chromosome 20. Now that's assuming I've mapped it correctly, and that's always it's something you have to kind of keep in the back of your head. But the limitation here is, are they related to me on the maternal side or on the paternal side? And the answer to that, of course, is yes. Right? They're related to me on one side or the other, but using the overlay feature alone, it doesn't tell you whether it's maternal or paternal. So often, all you can do is narrow it down to one line or the other. But if you can bring in any sort of shared matching, you'll be able to say this is a maternal match or a paternal match. And in this particular example, when I look at the shared matching of those 5 individuals, I do have a paternal match in that list. So I have a hypothesis now. This is likely to be a paternal group, and so that suggests our relationship is through John and Jane Smith Doe. So now I can go back to my cluster, and I can say something like, this is the Jane and John Smith Doe cluster that I'm currently working with. Right? Now, I can go through each of these clusters and try to determine what the common ancestry is. What I like about this approach, however, is that it helps me characterize 5 matches all at once. Rather than going through each one individually and using the chromosome browser in this approach, which I could do, this is a much easier process to label those matches. Now, what would I do when I make this discovery? Well, first of all, it is very important that you keep good notes, right? And you can do that however you want to do that. One thing you definitely want to do, however, is you want to put in the notes field at MyHeritage what you've learned about this match. Every match has a little notes field up in the corner with that match, and so you can put in the notes field. Because what happens if you don't keep good notes like this is you get to make this discovery over and over and over again, right? And that's not an efficient way to keep track of your matches. The other thing is, now what happens when these 5 matches show up in another shared match list? Right? So if I check with a new cousin and up comes one of these 5 matches I've already characterized to the John and Jane Smith Doe cluster, now I can hypothesize they're also related to the Jane and John Smith Doe cluster. Okay? And so it's you could think of it as ripples in the pond. The better notes you keep with your matches, they're going to show up in new shared match lists. And then they show up in shared match lists and so on. And it allows you to do this over and over again and create bigger and bigger circles of these shared matches. So chromosome mapping can be a really powerful way to both organize your known matches in a nice visualization that's fun and it's a nice way to organize it. But perhaps even more importantly is that we can do all this work to create a map. We can actually characterize new matches. We can work with these auto cluster shared match groups, and there's even more applications that are potential that we could use with these maps. So if you get started creating a map today, then down the road, you're going to be able to take advantage of that more and more as new tools and new options and new capabilities come along. So I will stop there and see if there are any questions about chromosome mapping using MyHeritage. Any questions about either the process of mapping or utilizing the map that you've created? Yes, right here.
Can you also see the names of the people?
So she's asking these clusters that I made, can I also see the names of the people? And I should have mentioned that here is, well, I lost the presentation. Remember when I talked about going down and seeing that table that table is what we want to see. So that table contains the names. I just blocked them for privacy. So if we look here, underneath that red box, those are the names of the matches. Yes. Yep.
Does it also, is it important how big those mini centimeter, whatever, how are they called?
Does it matter how big those centimorgans are?
Centimorgans.
It does in a way, because the bigger they are, the closer the relationship. So if you can work with bigger pieces of DNA, you're going to be better off, because you're going to have to presumably do less work to find the overlap between your trees. Small pieces of DNA, say 10, 15 centimorgans, can be rather old, and actually be really difficult to figure out what ancestor you share with a small piece. But if you're talking about a piece of DNA that's 50 or 75 centimorgans, presumably that relationship is much, much closer, and so you're going to have an easier time working with it.
And how much should the percentage be start looking? Because if you have 0.5%.
Well, sure. So how big should the piece of DNA be to start looking, or the percentage? I think of that, if we're talking about this particular analysis, the shared match clusters, for me, I think it's actually preferable to see the size of the cluster. Because with more people in a cluster, you're going to have more trees, larger pieces of DNA potentially. So I would start right away with this red cluster. That's probably going to be, on average, the easiest one for you to find common ancestry or a large piece of DNA. The further down the list you go, as you can see, there's fewer people in each of those clusters. That's going to be a lot harder, because then you'll run into a cluster where no one has a tree. The pieces of DNA are small. It's going to be really hard to work out the ancestry, unless you can do things like what we just talked about. In that example, even if the relationship is far back, you can still narrow it down to a potential line using your chromosome map. So, time for one more question. Yes.
I am really excited to hear that there's such a thing as a chromosome mapping software.
Mm-hmm.
So is that like something you buy somewhere, or is it something that you find? I mean, it doesn't come with Microsoft, right? I mean,
Yeah, no.
Where do you get this
Microsoft
stuff?
has not yet implemented a chromosome mapping tool. So there are a couple of different chromosome mapping tools available. So it used to be, again, that we would do it all in spreadsheets. Not so much anymore, because they're just unwieldy. But the main chromosome mapping software is DNA Painter. That tends to be the main one. You know, I don't know if there's going to be any competitors in the future, but that's certainly a tool that allows you to organize your matches. Yeah. Any other questions? I think we have time for, in the back there.
So let's say your cluster 5, let's say you had an additional match that had 2 segments, right? For whatever reason, mathematically, it's assigned to that one cluster, but that other segment is not accounted for.
Yep.
And then let me have a comment. One of the great features of the auto cluster is when you type the notes in,
Yep.
the next time you download it, the notes show up.
That's exactly right. So he's just pointing out that when you keep good notes, like we talked about, the auto cluster will pull those notes down and include it. So it would be listed, for example, on this table. You would see notes in the notes field. It's cut off there at the end, but you would see somewhere where you might have said, based on shared matching, the Cowell family or the Johnson family or whatever it might be. Now, when I talked about sharing a segment of DNA on chromosome 20 with this person, on this group, I focused on this segment because it was the largest segment shared by the group. A couple of the people in this group did share another segment somewhere else. But for my purposes, having this one segment was good enough to go to a map and identify where those pieces of DNA came from. Now, what I would use those other pieces for example, is sort of a second check. For example, if I say this aligns with a certain family based on my map, the segment here on 20, now I can go to the one I found, the little piece on chromosome 5, and maybe the little piece I found on chromosome 10, and I can say, does that also overlap with that same family? If not, then maybe there are multiple relationships involved. So you can use those other pieces but often what I find is many clusters, at least many people within that cluster will share a single piece, and I can use that then to do this type of assignment. And with that, we will end there. Thank you very much.